It's pretty incredible when you stop and think about how a simple bacterium can be so, well, smart. They don't waste a single ounce of energy, and a huge part of that is their genius ability to turn genes on and off like a light switch. Today, we're diving into one of the most famous examples of this, the lac operon. And trust me, it's more than just a cool biological circuit. Figuring this thing out was a total game changer for how we understand the code of life itself.
All right, so here's our roadmap. We're going to start with a why, the cell's big energy problem. Then we'll look at the brilliant solution, this thing called an operon. We'll meet all the key players inside the lac operon, and then we'll watch it work in two different situations. And finally, we'll wrap it all up by looking at the pure, simple logic behind this masterpiece of evolution.
So, first things first, why even bother turning genes on and off? Well, think about your own house. You wouldn't leave every single light on, the TV blaring, and the oven running 24/7, would you? Of course not. That's a massive waste of electricity. A cell feels the exact same way about its energy. It's all about efficiency. It only makes what it needs right when it needs it. Okay, let's set the stage. Our main character today is the bacterium E. coli. And E. coli's absolute favorite food, its go-to energy source, is glucose, simple sugar. But what if there's no glucose around? What if it's floating in, say, a sea of milk, and the only food available is lactose, or milk sugar? The cell has a choice to make. Does it go to all the trouble of building the tools to digest this new food? This is where nature gets really clever. Instead of having a whole bunch of separate on-off switches for every single tool it needs to break down lactose, the bacterium bundles all the necessary genes into one neat little package. It's a package deal, all controlled by a single master switch. And this brilliant little unit is called an operon. So what exactly is an operon? It's basically a stretch of DNA that has a few things. It's got the genes for the tools themselves, the proteins that do the work. And it also has the control sections, the bits of DNA that actually turn the whole production line on or off all at once. All right, time to pop the hood on the lac operon itself. Before we can see how this machine runs, we've got to meet the cast of characters, all the individual parts that make it work. So here they are, the key players. We've got the promoter, which is basically the start line. We have the repressor, which comes from the I gene. Think of this as our security guard. Then there's the operator, the O region, which is the actual switch. And finally, you have the structural genes Z, Y, and A. These are the blueprints for the actual lactose-busting enzymes. Let's talk about that repressor for a second. It's a protein, and its one and only job is to get in the way. It's a security guard that stands right on the DNA production line and physically blocks the factory machinery, a molecule called RNA polymerase, from doing its job and reading the gene blueprints. And where does the security guard stand post? Right on that little stretch of DNA called the operator. The operator is the specific docking site that the repressor protein is built to grab onto. This spot is the on-off switch. When the repressor is sitting there, the whole system is switched off. Okay, time to see this in action. Scenario 1, our E. coli is in an environment with zero lactose. There's no point in making lactose-digesting enzymes if there's no lactose to digest, right? So the system's default state is off. Now let's trace this on the top half of the diagram. The I gene makes a repressor protein, and you can see it there clamped down tight onto the operator, or O region. It's a physical roadblock. The RNA polymerase wants to go and read the Z, Y, and A genes, but it just can't get past the repressor. It's blocked. The end result? Simple. No transcription happens, which means no enzymes are made. And most importantly, precious energy is saved. The cell is being smart, not building tools it doesn't need. But now, let's change things up. Scenario 2. What happens when lactose suddenly shows up on the scene? This, my friends, is where the system's genius really shines. The very molecule that needs to be broken down is the key that turns the whole machine on. So check out the bottom half of the diagram. A molecule of lactose, we call it the inducer, floats into the cell. It finds our security guard, the repressor protein, and binds right to it. And this is the cool part. That binding action forces the repressor to change its shape. It can no longer hold on to the operator DNA, so it just bops off. The security guard has been neutralized. And with that repressor gone, the roadblock is cleared. Suddenly, the path is wide open. RNA polymerase can jump onto the promoter and zip right down the DNA track, reading the Z, Y, and A genes. The factory is officially open for business. So what exactly are we making? 3 really useful enzymes.
The Z gene gives us beta-galactosidase. That's the heavy hitter that actually chops lactose in half. The Y gene makes permease, which cleverly builds a little doorway in the cell wall to let even more lactose in. And the A gene makes transacetylase, which plays a supporting role in the whole process. So let's just take a step back and appreciate how unbelievably elegant this is. It's a perfect solution to an energy problem. The system is off by default, saving energy, and it only gets turned on when the very substance it's designed to process is physically present. It's brilliant.
This table just lays it out all out perfectly. No lactose, the repressor is active, bound to the operator, and transcription is off. Lactose present, it acts as the inducer, knocks the repressor off, and boom, transcription is on. It's a beautiful, direct cause and effect switch.
Now, this type of control system has a formal name. It's called negative regulation. And you might wonder, why negative? Well, it's because the thing doing the regulating, that repressor protein, is what actively turns the genes off. The system only gets to be on when that negative influence is taken away. Okay, so let's wrap this up. What are the big ideas to take away? 1. Gene regulation is all about saving energy. 2. Operons are nature's way of creating a package deal for genes that need to work together. 3. The lack operon is inducible, meaning the food source itself flips the switch. And 4. It's the classic example of negative regulation, where the default state is off until a blocker is removed.
The lack operon is beautiful because it's so simple and logical, but it really makes you think, doesn't it? If this is how a tiny single-celled organism manages just a few genes, what does that look like in something as complex as a human being, with trillions of cells? How do we orchestrate tens of thousands of genes to build a brain cell here and a muscle cell there? The basic principles are the same, but the layers of complexity on top, well, that's a whole other story.